Hi, everyone. This is Inga Hansen, editor of Medesthetics magazine. Thank you so much for joining us today. We're going to go ahead and get started with today's webinar. If you have any questions during the presentation, please type them into the box on the right-hand side of the screen, and we will answer them at the end of the webinar. Today, we'll be discussing how to incorporate effective natural skincare products into your private medical practice and how these natural products can benefit both clinical and aesthetic sides of a dermatology practice. Our presenter is Carl Thornfeldt, MD, FAAD, a dermatologist, researcher, and medical director of CT Derm. He's also the founder of Episciences, manufacturer of the Epionce skincare line. Welcome, Dr. Thornfeldt.
Good afternoon, and I guess it's good morning wherever we are in the country. So thank you so much for inviting me to speak today. So we're going to be talking about a variety of subjects here. Let's focus initially on consumer trends. Natural beauty and skincare products are desired today, but there's also demand for efficacy. They want reduced or no irritation to the products, no skin sensitivity, nor any allergenic type of reactions. So the clinical needs are basically wanting clinical efficacy and safety, but this needs to be done by scientific validation. And that's important because patients or clients expect us to be a clearinghouse for information to determine what's going to be effective and safe and to create the best regimen for them. It's important to realize that all clinical trials are not equal, and we'll be addressing that. So both the consumer demand and the clinical needs are equally important. The goal of this presentation is to walk through a strategy to address both issues equally, thereby keeping your patients happy and you doing the right thing as a skincare professional and as a physician. So what we need to think about are specific situations here and components of this whole process. One is the pathophysiology. In other words, the foundational abnormalities or the root causes that produce what we see as a skin disease or a skin condition, including skin aging. We need to know the mechanism of action of the proposed therapeutic product. Are these active ingredients physically, chemically, and photostable in the finished formulation? Is the packaging protective and compatible with active ingredients? Is the product science based on in vitro or anecdotal data? The efficacy should be proven for the disease condition with a blinded clinical trial using the finished product. Then safety for the targeted age group should be proven also with the finished product. Today we will be getting into more detail on each one of these specific steps. So let's review the pathophysiology. That's one of the big words that you can wow your friends and influence people with. Basically means the root cause of the underlying process. 10 years of research revealed that there are two foundational abnormalities that drive not only skin aging, but many of the most common skin conditions. And these consist of disrupted, damaged skin barrier and excessive chronic inflammation in the skin and mucous membranes. No, it is not a deficiency of vitamin A, vitamin C, or alpha hydroxy acid. Now, these two abnormalities, as the root cause, have been published back in 2001. Dr. Elias and our research team showed that multiple chronic inflammatory diseases are characterized or flared by a damaged barrier. And then I published in 2008 a big review showing that chronic inflammation is the primary driver of extrinsic aging, but I can't take credit for that. Actually, Albert Kligman, who invented Retin-A, first published on that in 1988, showing that inflammation was a critical factor. So what are the diseases it's associated with? Not only photoaging and sensitive skin, but also a whole variety of dermatitis, atopic, seborrheic, psoriatic, and contact dermatitis, premalignant actinic keratoses and irritated seborrheic keratoses, polymorphous light eruption, which many people equate to as a sun allergy that they develop in the spring, melasma, ichthyosis, and pityriasis, exfoliative erythrodermal, which is a total body red scaly rash. And then there's the intrafollicular type of diseases as well. In other words, acne, rosacea, keratosis pilaris, where these abnormalities occur down in the pore and then will activate these diseases. So you can see that this is a critical foundational abnormality for a variety of conditions. Now, what are some of the triggering insults that layer upon and activate the pathophysiology? We're all familiar with ultraviolet light, but we now know that pollution more than doubles the ultraviolet light damage. So airborne pollution triggers a pathway that results in significant damage. It's been recently reported that infrared light and visible light can activate photo damage. Heat greater than 99 degrees Fahrenheit will do it. Smoking, climatic and seasonal changes, and cooking and grilling food can also activate these diseases. So how do we rejuvenate these two foundational abnormalities? Well, we need to optimize the structure and the function of the stratum corneum barrier by activating its repair. We need to rapidly repair the barrier after any kind of cosmetic or therapeutic procedure we do, such as microneedling, laser, chemical peels. We need to make sure that we do not induce excessive chronic inflammation, and that's primarily protected by post-op treatment. Then we need to mitigate activation of chronic inflammation. Now, the difference between acute and chronic inflammation is that acute inflammation is absolutely required for rejuvenation. We're not talking about modulating that. But usually 14 to 21 days after the insult, then you will get this uncoupling of matrix metalloproteinases. These are the bad boy enzymes that actually are primarily used early on for repair, but then as we move into chronic inflammation and the cell type changes, we see an uncoupling of the MMPs and their balancing inhibitors. So consequently, the MMPs become very active, and they actually produce destruction of collagen and elastin and a whole variety of cells. So they prevent us from getting optimum skin rejuvenation. That's why it's important for us to have proper timing for our procedures, and it's critically important to have proper post-op care and pre-op care as well, so that we can control those aspects. Now, it is a complex process. When the barrier is disrupted, so we're talking stratum corneum barrier here, 5 repair cascades are activated. Chronic inflammation involves up to 7 cascades in the skin. So it's not a simple process because the most common anti-aging products, the vitamin A, the vitamin C, alpha hydroxy acids, only work on 2 of those 7 inflammatory pathways. So complex processes do require complex solutions. Therefore, we went to the botanical arena. Why? Botanicals provide complex mechanisms that are often synergistic. There's about 1,100 herbs used in topical products in developed countries today. There's been recent articles suggesting that there's 100 or so. The reality is there's over 1,100 that are actually being used. The active ingredients are secondary metabolites. These are compounds used by the plant for storage or for protection. And there's about 40,000 of these that have been chemically characterized from 15,000 plant species. Now, many of these plants will have multiple of these secondary metabolites. And one of the most common secondary metabolites people are familiar with are the flavonoids. Now, keep in mind, flavonoids, though, have multiple functionalities. Flavonoids actually have 10 different mechanisms of action besides the antioxidant, which is most well known. And we want to harness all those to get the best effect. But there's over 200,000 species that have not even been systematically investigated for potential medicinal use. So in other words, there's a lot out there for us to work on. Now, what are some of the formulation benefits? First of all, these ingredients are stable because each extract has multiple active ingredients, and they contain synergistic functionalities and synergistic secondary metabolites. They oftentimes will have distinctive compounds that are not available in the commercial arena or not synthetically produced that may have some very interesting manipulation of various barrier repair or inflammation pathways, which is what we had discovered in our nearly 10 years of research. In addition, there are oftentimes other compounds that are there to enhance stability and delivery. So botanicals have very excellent use in products. However, it's like a two-edged sword, though, remember. So the mechanisms of action, it's important to understand as we go into this how we understand these things actually, these ingredients actually work. So the mechanism of action of the product. We need to determine the efficacy of each botanical extract. There's one company out there that says their products work by a certain way, but there's 11 different anti-aging compounds in it, and then they're saying, well, somehow believe that this particular molecule is the one that actually produces the benefit. Really? We also need to determine optimum delivery systems for the active ingredients because there's differences in chemical structure, architectural polarity, and we need to get it to the proper site. And we need to determine the therapeutic concentrations of each. Keep in mind, compounds are used in a bell-shaped curve, so you want the optimum concentration. If you have too much, you may induce a toxicity. If you don't have enough, it absolutely will not work. So, in a nutshell, efficacy occurs when you have a therapeutic concentration of the intact molecule delivered to the target cell or the skin layer in an active form without causing inflammation. So, what does that mean? Well, let's look at the green tea example. A lot of people tout the benefits of green tea, but there's not a single clinical trial documenting efficacy of topically applied green tea working in reversing photoaging. The Japanese showed many years ago that you need a 2% concentration to actually get to the base of the stratum corneum of the green tea. Any lower concentration never penetrated even through the stratum corneum. And yet in the United States, there's only one product that actually has a concentration above that, yet in the clinical trials did not show reversal of photoaging. So just because they're claiming efficacy of something that's been done in the test tube doesn't prove that it's going to work on living, breathing human skin. We need to determine the efficacy by performing in vitro release of ingredients into mucocutaneous surface. What does that mean? So, for example, the vast majority of lip products are very greasy. But when we actually looked at the compounds used in the mucous membrane to rejuvenate that, if it was put in a product that had more than 30% petrolatum, it was never released. It just wore off during the day and absolutely had no clinical benefit on the lip tissue. You need to perform permeation tests of the active to make sure that the active actually penetrates across the stratum corneum into the human membrane. Those are called Franz cell tests. We need to conduct in vivo studies, in other words, testing in little patches on living, breathing human skin to examine the mechanism of action. In other words, what does the ingredient actually do? Does it manipulate barrier? Does it manipulate inflammation? And then prove that function is performed with your final product. We need to then do a dose response trial in vivo to determine what's the best concentration of the actives. As I said, you don't want too much, but you've got to have enough to be effective. And then conduct efficacy, safety, and cosmetic acceptability patch studies or in vivo studies, and then do pilot clinical trials. Finally, the gold standard is perform a blinded controlled clinical study using a finished product that was conducted by a third-party research after there's been a institutional review board. In other words, a group of scientists evaluate it and say, yes, we think that this is a plausible theory and it may be effective. And by the way, we like the fact that you did a safety study. Of course, that's not required in the cosmetic arena. It's important to understand that not all extracts are equal. Potency, molecular profile, concentration, and stability can all be impacted in herbs by climatic and seasonal growing conditions, the time of harvest, how it's stored, the duration of storage. Then the extraction method. There are currently now 21 documented methods for extracting compounds, but the most common are the cheapest. One of those uses boiling water or steam. But think about it. Most vitamins and antioxidants are actually sensitive to the heat and are destroyed in this process. Another common way is alcohol extraction, but that destroys the lipids and many of the proteins and peptides. So the reality is that using these two most common methods, which is the vast majority of where the low-cost extracts come from, have little or minimal functioning of active ingredients after extraction. That's why I'm not surprised when they looked at cosmetics that claimed to have retinol. 40% had no measurable retinol. Another study looking at vitamin C, 83% had no measurable functioning vitamin C. A lot of it has to do with the extraction methodology. Now, it's also important that one botanical can have multiple functions depending on the extraction methodology used. We all know that safflower is a good moisturizer, but it's also important in producing barrier repair, anti-inflammatory effect, delivery of other molecules, and for epidermal differentiation. But each of these require different fractions. And so the way that you extract that method will determine what functionality you're actually going to get. So not all extracts are the same. But there are some proven botanical products. Syncatechins from green tea have been proven and have a prescription product for the treatment of genital warts. Lemon and eucalyptus oil has been approved by the U.S. Department of Agriculture as an insect repellent equal in efficacy to the DEET products, which are the most potent of the insect repellents. All right, so what's the clinical proof? Well, the reality is there are a number of herbs that have clinical proof. So to date, there's been 16 that have proven to firm the skin, 18 that provided photoprotective function, 25 of the 220 showed anti-inflammatory effect. 32 were proven to have a repair of a disrupted epidermis and stratum corneum barrier, and 44 herbs have been shown to either treat alone, but usually with a multiple number of herbs in the blend to actually reverse photoaging in a blinded controlled clinical trial. Now, there's lots of other claims made by many other products and many other ingredients. So, for example, there's over 8,000 antioxidants known. Only 40 have been studied in clinical trials, and half of those had no benefit at all. So you have to be very careful with claims that companies are making. Now, of the 220 herbs that have been proven in vivo to have anti-inflammatory effect, these are the 25 that are most effective. Because they modulate multiple of those 7 inflammatory pathways. And because these conditions that we see, the skin diseases and photoaging, are multifactorial, we need to modulate multiple of those inflammatory pathways. These are the 13 most effective barrier repair herbs. As I said, there's 5 barrier repair pathways. These are herbs that affect multiple of those pathways, even though there's 32 that have actually been proven to have some improvement in barrier function. Okay, so how do we get these active ingredients to the site, to the specific cell, to the specific layer within the skin? Well, that's another great name for you to use to wow your friends and influence people, called dermatopharmacokinetics. So that's the complex multi-step delivery process. That consists of 8 steps. You have to dissolve the active substance. You have to completely cover the epithelial surface. You have to partition the active substance into a fat-soluble lipophilic stratum corneum. Then that substance has to permeate throughout the entire fat-soluble and cellular stratum corneum. Then it has to go into a water-soluble epidermis. Then it has to diffuse throughout the epidermis and then go through the protein basement membrane zone to enter into the upper dermis, and then again enter cells through their fat-soluble lipophilic membrane. Finally, there needs to be clearance. In other words, there needs to be mechanisms there to destroy the compounds in the tissue that it is after it's been utilized, or have vascular transport away. So a very complex process. That's why it's not as simple as taking an herb and grinding it up and saying, here, use this, because it actually does work. The reality is the vast majority of them don't work at all, and we'll talk about that in a minute. So let's talk more about the ingredient formulations. There's an impact of how these ingredients are formulated in the manufacturing process that has an impact on product efficacy. Water-soluble antioxidants, peptides, proteins, cytokines, growth factors, probiotics, and vitamins are highly susceptible to heat and light damage that occurs during manufacturing and storage if they survive the extraction process. And as I mentioned earlier, we know the product effectiveness is impacted because, after all, 83% of vitamin C products have less than 50%, many down to 0% functioning of vitamin C. And 40% of cosmetics claiming retinol as an ingredient have no measurable amount of vitamin A in the formulation. So, what is the remedy for this? We need to manufacture a low ultraviolet light environment. We need to keep the product temperature below 50 degrees Celsius and use low atmospheric pressure devices. How often do private label manufacturers discuss with you that products they're going to make for you fulfill these requirements? They don't, because they don't do it in their standard manufacturing processes, and they don't want you deviating too far from the norm. But these are the important questions to ask in the manufacturing process. So let's look at the packaging, because the packaging needs to protect freshness, and that will improve your clinical results. So we want to ensure maximum product efficacy, then protective packaging is necessary. So you want to use black, dark blue or brown glass, metal, or multi-layer polyethylene glycol containers, or multi-layer polymers. And that will help keep out the oxygen, keep out the light that will destroy many of the active ingredients they particularly target, antioxidants. And that's why they're so difficult to formulate and so unstable. And incidentally, the first person that published on that was not me, but actually Sheldon Pennell, who was the inventor of the first vitamin C products. So packaging, as I said, it protects freshness, and that improves the results. Ideally, for tubes, you want one-way valve tubes to minimize the ingress of gases into the tube. You want freshness because when you're using multiple active ingredients, especially botanical ones, that they will have chemical interaction, and chemical interactions do occur. And unfortunately, some people would have you believe that somehow their product magically doesn't do that. How do we know that? Well, a number of years ago, we did a study and found that it took many months for private label products to even get to the physician's office, and that autooxidation was undoubtedly going on at that time. It occurs in all products. And so it's important to get product to patient, ideally within 4 months of manufacturing, to assure active ingredient function. When we do these IRB blinded clinical trials, we're required to have product no more than 30 days from the day of manufacturing. So if you have a product that's been sitting on the shelf for a long period of time, how can you tell a patient, use this product, we know it works? No, unless you have studied that product in that setting where it sat on the shelf for a year, no, you can't tell them that it works, because the odds are very great that it was destroyed by autooxidation because of interaction chemically between the various active ingredients. So product science then is based on in vitro or anecdotal data, or is it based on other hard science? Efficacy should be proven for the disease condition with a blinded clinical trial using the finished product, which I mentioned earlier. Now, just because an ingredient is in there does not mean it works. A matter of fact, the odds are very great that it will not. And in vitro data absolutely does not predict success of a therapeutic product. How do we know that? By the end of the 20th century, the FDA went back and looked at products that were brought to them with in vitro data. In other words, a laboratory test saying this works, so this should be an effective treatment for that disease. Only one out of every 350 potential medications actually became an approved drug. So the odds are very great that unless they've done clinical trials and proper product development and proper dermatopharmacokinetics, the product is not going to be effective, absolutely will not work, and you will have a disappointed patient or client. So this proves that the presence of any active ingredient does not ensure clinical efficacy. So what is the evidence-based for efficacy gold standard? In other words, how do we know that a product actually works? So we've talked about ingredients. Now we have made an ingredient where we have done stability and we've shown that the product is stable. But now, does it actually work? Do those ingredients in there impact patients' lives that they can see a visible change? So we use double-blinded, prospective, controlled clinical trials against a placebo or against an approved prescription product. This clinical trial is then conducted by a third-party contract research organization after the protocol has been approved by an institutional review board. In other words, a group of scientists saying, yep, we believe this is a plausible and a safe thing to study. The trial must have enough panelists to prove statistically significant efficacy, at least 8 in the group. Okay. Now, one of the concerns that has been raised is people said, well, you need big clinical trials to show statistical significance. Actually not. Back in the 80s, it was documented that small clinical trials going up to 25 to 36 patients was as statistically significantly in understanding market acceptability. In other words, if you could not show that there was a statistical significant superiority of your test product over a placebo or a prescription product with 12 patients, it would not be accepted in the market. So you could do hundreds and hundreds of patients, but it does not increase the market acceptability because the differences patients see are not enough for them to be motivated to buy the product. So clinical trials in this case, where we're primarily at marketing stories, we want to make sure that they actually are proven in small clinical trials. Now, the FDA has set the bar for cosmeceutical efficacy, and that is sunscreens. They require a sunscreen has to have at least a 20-panelist clinical trial to prove efficacy. We physicians demand evidence-based medicine for prescriptions and for devices, and we need those approvals before we're going to use them in a clinic. Okay, those prescriptions is called a new drug application, or an NDA. Unfortunately, takes years and costs hundreds of millions of dollars. The device approval for certain levels of lasers and others is the 510K. And those are processes approved by the FDA that we then use in our clinic. So why should we have a double standard? Why should we be selling and recommending skincare and anti-aging products that have no safety and no efficacy trials? In my mind, that's a moral, ethical issue, and that's a conflict in and of itself, because we as skincare professionals, if there are products that have proven safety and efficacy, why would we withhold those products from using them on our patients to produce the best clinical result and provide them with the healthiest skin possible? Okay, so let's look at skincare products that meet these standards, because there are a few that do that. There are a few cosmeceuticals that meet these standards that have been developed and tested against prescription or other anti-aging products. So only 2 companies, however, have conducted trials against prescription products. One of them was then acquired by a major pharmaceutical firm. The other one is still in operation. There were 3 companies that did trials against other anti-aging products. One company did a test against tretinoin 0.02% for anti-aging, and they showed equal safety but not quite as good efficacy. There was another product that was a peptide product that was tested against glycolic acid, which nobody doubts has some measurable benefit in reversing visible photoaging. But then there's a third company that has conducted 16 blinded clinical trials, 10 of which were against prescription products, and those particular products were superior, statistically superior in efficacy and in safety. So let's review those particular products. So there was an 8-herb product with barrier repair and anti-inflammatory lotion and cream, and that was tested against emollient tretinoin 0.05%, in other words, the Renova 0.05%, and their lactic acid moisturizer in a 6-month trial. Clinically, there was little differences between on initial examination, but with biopsy, it showed that the herbal product, which consisted of Epionce Renewal Facial Cream and Epionce Lytic Lotion, was nearly twice as good as the Renova regimen, including their lactic acid moisturizer, in reducing in stimulating epidermal glycosaminoglycans. Now, why is that important? Well, these GAG fragments actually diffuse across the basement membrane zone into the dermis and activate fibroblast synthesis, as well as activate epidermal keratinocyte synthesis. So very important. So there was a statistical significant superiority of the herbal product over tretinoin, the gold standard. But then when dermal density was evaluated with micro-ultrasound, it showed that the dermal density was more than twice as good with the herbal product than with Renova. In other words, the Renewal Facial Cream plus the Lytic Lotion was double the efficacy in the dermis over the repair of the Renova and lactic acid. So when new companies have come out in the last couple of years and are talking about a retinoid alpha hydroxy acid product, so what? That was beaten 12 years ago by this particular regimen. There was a 10-herb cream that was then tested against itibanone. So that 8-herb cream was taken, and a potato peptide and a castor bean-derived azelaic acid was added to it. And this was tested against the most potent antioxidant on the market, called itibanone, which when it hit the market, everybody said there's no antioxidant as powerful as this. And yet, comparing this intensive nourishing cream, Epionce intensive nourishing cream, with the prescription strength itibanone, the intensive nourishing cream was 3 times better in getting rid of shallow wrinkles and highly statistically significant. And its effect on dermis was more than double. Also statistically significant superiority, again showing this concept of barrier repair and inflammation modulation is better than not only the retinoids, but also much better than the antioxidants. Then a 9-herb serum, which contains all 23 vitamins required for epidermal function, including, and by epidermis we're referring to the stratum corneum, the major part of the epidermis body, and then the base membrane zone. That particular structure, that epidermal structure, requires 23 vitamins. And so a product was designed called the Epionce Intense Defense Serum, and a delivery system designed to deliver all these vitamins throughout the entire epidermis, and had significant results. One clinical trial compared it with a multiple growth factor product, and the other with a vitamin C antioxidant blend in these 2 different clinical trials. In 1 week, there was highly statistical superiority in reversing fine wrinkles, indicating the importance of nutrition in our skin. If we don't provide the vitamins that the epidermis needs when we call upon it to rejuvenate, it can't do it. You've got to provide the proper nutrients at the right concentrations, deliver them throughout the entire epidermis to produce the benefit. So, as I said, highly statistically superior at 1 week, but at 8 weeks, 57% reduction in fine wrinkles, 50% reduction in closed comedones, 46% reduction in fine lines, 36% reduction in dyschromia without a single tyrosinase inhibitor in that, and 32% reduction in coarse wrinkles, which tells us the importance of controlling inflammation and how active inflammation plays a role in activating the hyperpigmentation. But what was most important, 78% of the patients preferred the intense defense serum, and a number of them were having active dermatitis reactions to the other products, but they wanted to stay in the clinical study so they could get the free bottle of the IDS at the end of the trial. Then another clinical trial with a 3-herb oil known as our priming oil, which contains meadow foam, safflower, and coconut oils, over the Epionce medical barrier cream, which consists of meadow foam, rosa canina, white water lily, and onion, combined with the 3 key physiologic barrier lipids, which are cholesterol, ceramide, and fatty acid. We compared that regimen with acetic acid soaks and Aquaphor for 4 days after laser resurfacing. And the goal here was to reduce the risk of infection and post-treatment hyperpigmentation, which occurs a year later, and 2 clinical studies showed a third of the patients that were treated with these procedures, in fact, had post-inflammatory hyperpigmentation. 2 different resurfacing devices were used, a Cyton laser and also a CO2, as well as a superficial chemical peel. And it was found that at day 2, there was highly superior statistical significance of the 3-herb oil over the 4-herb cream compared to the Aquaphor acetic acid soak. And there was also highly statistically superior in reducing transepidermal water loss, which is the measure of barrier function, skin barrier function. So 100% transepidermal water loss means no barrier function at all. 0% transepidermal water loss means the barrier is 100% intact. Now, at day 4, both regimens were equal and had returned to baseline, indicating there was complete healing at 4 days on a functioning level from these particular products. But what was most important too was these trials showed for the first time that using post-treatment care had a great impact on photoaging 28 days after the procedure. So the herbal blend and the traditional were used for 4 days, and then no treatment for another 24 days. And then the patients were reevaluated. At day 28, it was found that there was a highly statistically significant improved global facial firmness with the herbal program. And there was also statistically significantly improved forehead and perioral wrinkles, jowl sagging, and evenness of skin tone, showing that 4 days post-op care is critical to optimize your results in reversing photoaging, no matter what device you use or if you use chemical peels, but post-care is absolutely critical. So when companies tell you do not use a particular post-treatment device or post-treatment care regimen, that's generally because they never bothered to spend the money to do the study to prove it, but don't heed that. We clearly showed that it's absolutely critical. And this is not just me, but other researchers have also documented this as well, and big reviews were published in December of 2013 showing conclusively that there's no place for air drawing after procedure, and that, in fact, petrolatum is far better than Aquaphor because more than half the patients with Aquaphor use will have persistent erythema. Okay, another product was a 7-herb cream called the Renewal Calming Cream, and that was shown to be effective in eczema therapy. It is an FDA over-the-counter monograph treatment for eczema, and what those FDA over-the-counter monographs are, these are documents that the FDA says if you use certain ingredients for certain conditions in certain concentrations, you can claim it works for that. So we used a therapeutic concentration of colloidal oatmeal. But the interesting thing is that the range that the FDA allows for that oatmeal concentrations is 10,000-fold. And we found a concentration that was highly effective in reversing symptoms, but did not allow activation of autoimmune diseases nor of gluten sensitivity type of processes. And so that's the concentration we used, which still was much higher than the minimum concentration that the FDA allowed. So how did it work in patients? So these were patients suffering from moderate eczema, and we found that in day 8 there was a 58% decrease in itching or pruritus, 58% decrease in scaling and dryness, and a 39% decrease in redness or erythema. But by day 22, 81% decrease in pruritus, 68% decrease in scaling and dryness, and 54% decrease in erythema. Are those big numbers? Yes, they actually are, because we know that several prescription products have significant benefits. So, for example, Eucrisa, the most recently approved drug for the treatment of eczema, had 14.7% improvement by day 8, and at day 29 had a 31% improvement on their global score. So here our numbers, even though we didn't do a comparative studies, our numbers were substantially more effective, and in fact that these results were better than what's been published for tacrolimus as well. So the two prescription products that are non-steroids, we were safer and more effective in both. All right, then the most recent product is a 6-herb gel, which is an OTC monograph for acne containing 2 OTC active ingredients. And we compared this with clindamycin benzoyl peroxide. And let me get to that. All right, so and this particular purifying spot gel contains spearmint, coconut, date, paprika, sunflower, and acacia to make it a colloidal sulfur suspension, and then 2 FDA over-the-counter monographed acne drugs, colloidal sulfur and resorcinol monoacetate. So it didn't smell, it didn't stink, it didn't stain, it didn't induce hyperpigmentation. But what was important was how quickly it worked. Here it was doing a test against the prescription gold standard, clindamycin and benzoyl peroxide, which was actually shown to be equal to the tretinoin benzoyl peroxide products that have hit the market, but statistically superior to plain tretinoin, to plain benzoyl peroxide, to plain clindamycin. And yet after 3 applications, purifying spot gel achieved statistical significant superiority in resolving the lesions. And at day 6, we were highly statistically superior in reducing lesions, whereas the clindamycin benzoyl peroxide only reached statistical superiority. So at day 1 and at day 6, our performance was faster, more effective, and without irritation reactions than prescription clindamycin benzoyl peroxide, the 10th prescription product that Epionce beat. All right, let's look at safety. Safety in skincare. So the Cosmetic Toiletry and Fragrance Association recommends a 50-panelist repeat insult patch test to determine whether a product induces irritancy or sensitization. But very few cosmeceuticals actually do this. When I walked the floor at the American Academy of Dermatology several years ago, of the 38 cosmeceutical companies, only 2 had done these safety studies. And yet, these safety studies are fairly low cost, all less than $4,000. I believe safety is a moral ethical issue for the prescriber or the retailer, because after all, patients are coming to you for products, for prescriptions, and procedures that have shown to be effective and safe. But we do it because it's the right thing to do. So these are the topics that we've addressed today. So what's the benefits of our Epionce in clinic? We address the 2 root causes of skin aging and skin conditions. We have original bench research for the botanical ingredients. That's why we don't use other common ones, because the common ingredients out there like teas and soy didn't work well enough. These herbs we use are much better. The formulations are done with proper product development. The packaging enhances the product efficacy, and we've done 16 independent clinical trials, of which 10 were against prescription products. And finally, we've proven safety for you. So this is a botanically based skincare line with proven efficacy and no irritation or sensitivity reactions. Okay. So a quick case here from my clinic, a 39-year-old mother of 4 with Asian skin who suffers from 10 to 15% body surface of eczema plus sensitive skin. She sleeps poorly. She doesn't feel good for many years, has never been clear of her visible disease nor itch-free. She has suffered multiple reactions to oral antibiotics and oral medications, including stretch marks from topical corticosteroids. And yet on my exam, she had crusted, weepy, scaly patches on her neck, scalp, arms, hands, and feet. The bacterial culture revealed staph bacterial infection. But one of the problems had been is that not only did she react to many oral antibiotics, but she also found that topical prescription products universally burned her skin, whether it was a topical corticosteroid cream or whether it was Protopic 0.03% ointment or whether it was moisturizers or topical antibiotics. They all would produce a burning sensation. I treated her with renewal calming cream 3 times a day and the Protopic 0.1% ointment only for nightly, and did that for 6 weeks. Then used the renewal calming cream twice daily for 3 months and then once daily. I gave her 15 days of an oral antibiotic and put her on a supplement called quercetin, which is anti-inflammatory and antihistamine, and she took that twice a day for 4 months. By 3 weeks, she was completely itch-free. She had no reaction to Protopic. Remember, at the lower concentration she had reacted to, and she got full night's sleep for the first time in years. Her rash was completely clear with normal appearing skin at 6 weeks. And importantly, most importantly, she was clear and itch-free for 5 months, even as we moved into the winter. So in other words, knowing how to use these various treatment products that Epionce has designed by understanding the underlying disease and producing proper formulation, you can impact people's lives for the better. And after all, isn't that what we are all called to do? That's why we chose being a skincare professional, to improve the quality of life of our patients, and these products do that. Thank you very much for your attention today. If you have any questions, you can contact us at epionce.com.
Yes, and we actually do thank you so much, Dr. Thornfeldt. We actually have questions coming in right now from some of the attendees. And if for any reason we aren't able to get to your question or you think of something later, as Dr. Thornfeldt mentioned, you can reach out to Epionce or to me here at Medesthetics, and we will make sure we pass that on. Someone had a question about the post-procedure care, saying that they currently use either Aquaphor or Stratacel for RF microneedling. So do you recommend medical barrier cream as a better option?
Yes, we actually have a kit for post-op care that uses those 2 products. It has the medical barrier cream and the priming oil and the firming mask that has been shown to be, that's what we used in this clinical trial here. And so we know that it works. The problem with Aquaphor is since I'm a referral-only derm, they have to see primary care first. I see a lot of the messes. And it's been published that 53% of the patients with Aquaphor will have persistent erythema, 38% will have persistent edema, and a lot of the reactions I see are from that later. Yes, in the first 24 hours, because of its occlusive nature, it has some benefit. But when you actually look at what the ointment does by 8 hours after application, you only have closed the barrier about 60%, whereas our barrier technology has closed the barrier 100% by 2 hours after the procedure. Now, keep in mind, if you don't use anything and just let it close on itself, it may take as long as 14 days for that barrier to completely clear and return to normal functioning.
And now, so your product that has the botanicals can be used immediately before the reepithelialization.
Yep. Absolutely. It speeds it up, and especially during the reepithelialization, during that time, the environmental factors that are pro-inflammatory and pro-infectious are penetrating in 200 to 1,000-fold greater.
And we had another question about post-procedure. I'm not sure what the abbreviation is, but someone asked, would you recommend IDS with microneedling either during the treatment or post-care?
We have protocols for these different procedures at Epionce, so it's best to get those protocols. One thing about the IDS, remember you need to use the right tools for the right thing. The IDS is focused on epidermis and provides all the nutrients optimized for epidermal function. Yes, it will have some benefit down in the dermis, but it's not a delivery issue here. We're trying to optimize. We have optimized function and delivery, and so punching holes through that can activate inflammation, that can increase metabolism and breakdown of these nutrients is not good either. So doing fractionated work and then putting IDS to have some effect, thinking that you're going to do a better job, is not the case at all. Our delivery system is so sophisticated and so quick, literally in seconds, it's penetrating through.
And there's a question that came in about the cannabinoids, which certainly have been getting a lot of attention lately. And the other question about whether you have looked at these or have any thoughts on the developing role of their use in skincare products and dermatosis.
The non-THC cannabinoids do have some anti-inflammatory effect. We do understand how they work. We don't use them in any of our products. And because the mechanism of action was not strong enough in any of the pathways that we were working on. But do they have a measurable impact? I'm sure they do. The problem is I haven't seen any clinical studies actually proving that it does work with those formulations. But certainly where there's a lot of people saying it works for them, I have no doubt that there's some measurable benefit. The question, though, is as with a lot of these products, as a doctor, this referral only particularly, so I take care of the more difficult patients. I've never had a patient come in and say, hey, can I get a little better? They all come in and they want the best treatment. They want to get as clear as quickly as possible. They want to stay clear for the prolonged period of time. That's why we did the basic research and understanding what drove the whole problem so that I can confidently prescribe for the patient. This is what will clear you the fastest. This will what put you into long-term remission and will keep you from flaring as the environment changes and your exposure to environmental insults change.
And we had a question about being able to, someone wanted to be able to go back and review the webinar, particularly to look at some of the natural ingredients that had proven efficacy. So just wanted to let people know that by the end of today, this will be up on medestheticsmagazine.com/webinar in our YouTube channel. So if you want to review it or share it with anyone, you can view it there. And it looks like those are all of the questions we have. So again, Dr. Thornfeldt, I want to thank you so much for all the great information, and thank you to everyone who joined us today.
Yes, thank you all for giving us the time. It's been a real pleasure, and as you can see, that there's a lot of complexity to this whole science, but that we can produce great results in patients' lives. Thank you.
Thank you.